This was a ton of information. So this chapter was different than the first two, whereas the first two were really focused on nutrition and putting that those concepts in the lens of nutrition. This one, because it was taken from a different kind of text, didn't really do that. So I feel like that's my role for you today, is to try to help put this in the context of the whole system, now that you understand what those individual parts were. Wait, is a macromolecule the same as like macros, macronutrients? Yes, they're totally the same thing. This is why they're called that. So macro just means big. So macromolecules are big molecules. They're often polymers. So there are lots of links in a chain stuck together. We'll talk about which ones really are and really aren't. But that's why they're called that. So you learned about the other macromolecule that most biology classes would cover is nucleic acids, which are the fourth macromolecule group. We skipped them because although DNA is extremely important, it's not really something we think about with regards to nutrition. Instead, we read about fat or lipids, carbohydrates, and protein. And I brought you this that we will take a minute to talk through. I know it looks really complex, to sort of demonstrate the interplay of these three different things and how they're processed in the body and the ways in which one might be transformed into another. So this diagram is trying to show the pathways for fat up here, for glucose, and then for protein. So they don't have dietary protein coming in through the intestine because we don't take a whole lot of protein out of the intestine. It's a small portion of what is taken up, and most of what we're taking up for energy purposes is fat and glucose. So fat comes in from the intestine through the lymphatics. So it doesn't come in through your bloodstream. The lymphatic system is the place where lipid and fat soluble things are transported. This is just showing you that fat is absorbed in the intestine, comes in through the lymphatic system, and then is processed in the liver. So from the liver, fat is going to enter into the cellular respiration process, which you'll learn about next week and the week after. And it will be processed to create ATP for energy. And the molecule will then be turned into either ketone bodies. It can be transformed to stored fat in the form of triglycerides and then broken down into fatty acids. So on the other part, the other molecule that's coming in from the intestine is glucose. So glucose and other sugars are readily absorbed through the intestinal wall, and it's got a lot of different places that it needs to go. So glucose can go to red blood cells in the brain, and these are the two like most energy intensive kinds of cells that take up a lot of the glucose that's brought in, dietary glucose. So these two organ systems are taking a lot of glucose, and the rest of this glucose is often being processed in all the cells in the body through cellular respiration. So as glucose comes in, we're going to also convert it to CO2 and water. So you can see a little CO2 coming out over here, and that's just to demonstrate that glucose is brought into cells of the brain, processed to create that ATP for energy, and then CO2 is released. So that same process is happening in whatever cell glucose enters. And the last piece that's shown on here is that glucose can also be taken up by muscle cells. Okay, so this is trying to show you what's happening in the muscle, and this is where protein comes into play. So we store fat in our body in the form of triglycerides. We store carbohydrates in our body in the form of glycogen, and that's shown over here. But we don't store protein in the body for the purposes of storage. Protein is always used when it's in the body. But if your body has to harvest protein from some source because it's not getting enough dietary protein, or because you're not getting enough other nutrients from fat and carbohydrates, then where protein is taken is from the muscle. So if that happens, then muscle protein can get broken down into amino acids, and then those amino acids can travel to the liver. They can be broken into these other products that, if you follow these arrows that loop back around this way, and then can actually be converted into glucose. So muscle protein can be converted into glucose if your glucose levels are low. And that's why one of the most important functions of carbohydrates in your diet is to spare protein so that we're not consuming protein from our muscles instead. So consuming plenty of carbs means that your body won't need to do this. What have we missed here? A couple other things on here. Here's your adipose tissue. So we said that we store fat, and we store fat in adipose cells, and they're spread all throughout the body. And then here's our stored glycogen. So this is the carbohydrate, that carbohydrate storage molecule we'll look at more. And when there's lots of glucose in the bloodstream, that extra glucose can be converted into glycogen or into fatty acids. Okay, so somebody asked in the reading, is there a specific set of enzymes that's released into the digestive system every time you eat a meal? Which I thought was a really interesting question. So there is a suite of digestive enzymes that are available, and they're all being produced at different levels based on what it is that you eat and what the patterns of your diets are. So after you eat a carbohydrate-rich meal, then there will be signaling molecules that are released in the stomach and in the intestine that are traveling to the bloodstream and communicating with the pancreas to see which of those digestive enzymes need to be released, the liver as well. So the amount of different enzymes will depend on what kinds of things you ate. So if you ate a really fatty meal, you're going to release a lot more lipase. So lipase, anything that ends with an ACE is an enzyme. Lipase means it breaks down lipids. Versus if you eat a really protein heavy meal, you're going to release a lot more proteinase enzymes. Okay, so I brought you another diagram. It looks just like this one, except it focuses in specifically on carbs. So I wanted to talk about that. So let's look at the regulation of blood glucose, because this underlies everything. So this was also alluded a little bit in the last slide, but glucose levels have to be maintained at a really specific level. So if your glucose level falls below about 70 milligrams per 100 milliliters of blood, then you will have symptoms of that. Mostly neurological first, because again, the brain is very glucose hungry. So if your blood glucose level falls, you'll feel tired or cranky or dizzy or any number of other symptoms. So on the other hand, your blood glucose level should not be too high, because that's also damaging. So if your blood glucose level goes outside of this nice little range, then the body is going to work to bring it back in. So if your blood glucose level goes too low, so if you've skipped a meal or if you're not consuming enough carbohydrates in your diet and your blood glucose level falls, then the pancreas is going to be triggered to release glucagon. And so glucagon is a hormone that's released from the pancreas that travels to the liver and releases that stored glycogen. So glycogen is our storage carbohydrate, and it can be chopped up. Glycogen is just a bunch of glucose molecules stuck together. So when it gets released, the blood glucose level rises, and that brings us back to homeostasis. The other thing that happens here that's not shown on this diagram is that there's a whole bunch of other hormones that are released as well. So when your blood glucose level falls, you're also going to get hormones released like ghrelin, which makes you hungry, and other hormones that are going to stimulate your appetite or stimulate your desire for sweet foods so that you can also feed yourself more. If your blood glucose level goes too high after you eat something really carbohydrate rich, then the pancreas is instead going to release insulin. So insulin does the opposite thing. Insulin is going to encourage cells to take glucose out of the bloodstream, not to put more back in. So any extra blood glucose is going to be sent to the liver to replenish that glycogen that was broken down over here. So there's this constant cycle of trying to make sure that there's plenty of glycogen in the liver when there's times of plenty, so that there's extra to break down when times are lean. The other thing that insulin does is it triggers all of the cells in your body to take up glucose. So the cells in your body can't actually consume glucose without insulin present, except for the brain and red blood cells, which do this independently because they need it so badly. So when insulin is released, it triggers all of the cells in your body to take up glucose. If there's still excess glucose, then the last thing that happens is that excess glucose can be stored as fat. So it'll be transformed to fatty acids and then stored in triglycerides. And all of those things together mean that your blood glucose level is going to decline. So this is what's happening in the background. What happens when we remove one of these nutrients, carbohydrates, largely from the system? So I want to go back to an image that looks like this. So this is showing just fat and glucose. And it's showing the typical things that are going to happen when you have plenty of fat and plenty of glucose in your diet. So just again, your glucose is going to go in lots of different places. It's going to be taken up by these hungry nutrients, going to be taken up by all the cells. We're going to release CO2. We're going to have some of it maybe stored in fat and some of it stored in glycogen. And because we have high blood glucose, insulin is being released. So this blood glucose is being able to be brought into all of these different cells. So if that blood glucose is not there, so in this one, you have blood glucose coming in from the intestine because you have eaten carbohydrates. If we take that part away and we look at this diagram instead, we can't not have glucose in the system. And so this is the way that system will adapt to create more glucose in the system. So instead, we're going to create glucose from glycogen, and we're going to mobilize muscles to create lactate, which also can go to create glucose. This process is called gluconeogenesis, which means creating glucose from something else, making it new. And glycogenolysis means to break down glycogen. So both of those things are going to happen to increase your glucose levels so that you can feed these needy cells. The other thing that can happen, hold on, let's go back, because this one doesn't show it, is that we're also going to have ketone bodies released if your diet is increasing the level of fat while decreasing the level of glucose. So some diets are just limiting carbohydrates. Some diets are limiting carbohydrates and purposely increasing fat. So depending on what the balance of low carb diet that someone is experiencing, it will shift the way that their body is going to respond. But if there's plenty of fat being brought in, which is often what's happening if you're replacing carbohydrates, then that extra fat can also be processed to create ketone bodies. So ketone bodies act as a sugar. They act sort of like a backup glucose source, and they can be utilized in similar ways by the brain and other glucose hungry organs. So those ketone bodies will circulate inside of the body instead of glucose. And it can feed those cells and give them the energy they need in this time of glucose starvation. So you have lots of questions about this. The big question that everyone wants to know is this safe? Is it a good idea? Does it help one lose weight? Does it help with other health challenges? Is it sustainable? So there have been not that much study on the newest forms that like paleo that are adding fat and glucose. Those have not been studied as much because they're relatively new, but low carb diets have been around since the 70s, and they started with the Atkins diet in the 70s. So there's been a lot of work comparing low carb diets to other forms of diet for weight loss. So for weight loss purposes, low carb diets are very effective in the beginning. So compared to other typical diets that folks might try out, like Mediterranean diet or just a low calorie diet or a low fat diet, low carb diets tend to have greater weight loss in the first year. However, they also tend to be less sustainable because they're a really drastic change to the diet. And so it's very difficult to avoid carbohydrates. And so many folks can't persist on a low carb diet over time. So one of the things that happens with dieting overall, and I should have brought you this slide, but I forgot, is that anytime you diet and are reducing your total calorie intake, you're also damaging your metabolic rate. So in week 5, you're going to read more about our metabolic rate. One of the things that happens if folks can't persist on a certain diet and they've been on it long enough to reduce their metabolic rate is that when they go off of the diet, their weight regain will be greater than the rate of weight loss. And so the end result for most folks who try a low carb diet for weight loss is a yo-yo, and they don't tend to have sustainable weight loss. The diet that has been shown to be most sustainable over time and most healthy every single time as the studies are done is the Mediterranean diet. So the Mediterranean diet offers a really balanced diet, and any weight loss that's happening happens much more slowly and is more sustainable because it has lots of balance in the food groups. So folks tend to be able to persist on it long term because they're not restricted to one kind of food group or another. The place where low carb diets have the most importance, and I brought you this really cool visual abstract, are in regulation of diabetes. So I love this. This abstract just came out last month. And to me, this is like a scientist version of an infographic where they like try to convey all of the science in this really cool paper into one image. So let's break it up a little bit. So this one was not looking at weight loss. It was looking at the potential to cause diabetes to go into remission or to improve diabetes symptoms. And so this was a systematic review, which means that they did not conduct their own like clinical trial, but instead they looked at all of the data that had been collected on clinical trials, and they combined all of these studies together to create this much larger data set that they could analyze, giving them more statistical power to see potential effects. This is a really powerful way to do science. And if you're ever trying to answer these like really big questions, like does a low carb diet work? Looking at an individual study is not the way to go. And unfortunately, that happens a lot in the media, that there will be a study that comes out that relates to something like this, and everyone will say, oh, this one study found this thing, that's so cool. But that's one study. And so instead, if you're really trying to make informed decisions, you should look for this kind of work. So secondary sources and systematic reviews and meta-analysis, take primary sources and stick them all together. So this week, you're going to find a primary source and for your group task, and you'll see the sort of differences between that source and what they've done, and then what this has. So back to this. This study ended up looking at 23 studies that fit the qualifications of concluding the right kinds of information that they could do this analysis, and increase their sample size from studies that are usually really small to having over 1,300 participants. And what they were able to look at were the effects of low or very low carbohydrate diets on remission, with or without medication, and whether or not there were any adverse effects or weight change. So low or very low carbohydrate diets are defined in these specific ways for the purposes of scientific studies. A low carb diet is typically what someone might be doing if they are on an Atkins diet, and a very low might be one of the more extreme versions where they're trying to eliminate carbohydrates entirely and go in like the keto diet. So these charts down here are showing you the difference in risk for groups that were either experimental studies where they were not on low carb diets versus low carb diets. So the risk difference, this handy little diagram is showing you that if this mean spot right here is closer to the green side, that means a low carb diet has improved this condition. And if something is closer to the pink side, that means a low carb diet has negatively impacted this condition. So in the end, what they can see here is that there are a few things that were effective. That reducing this HbA1c level, which is a marker of glucose, of insulin resistance and blood glucose levels, did have important changes. But they make a note down here that even though these were statistically significant changes, the clinical importance of those differences were really small. So there were differences, and that is a reason why someone might explore using a low carb diet to manage diabetes, but the differences were small. For some of those folks, though, they had remission of their diabetes. So I would imagine that these would be folks who were probably earlier in their progression in diabetes, because diabetes is known to be well managed by a diet if it's addressed really early on. The difference was lost, however, when they asked if those folks were so far into remission that they could no longer take their diabetes medication, because for most folks that was not true. So they had a great reduction in symptoms, but they still had to have some diabetes medication or management of their blood sugar over time. They also did show that there was a weight change, that there is weight loss that's associated with low carb diets as well. Yeah, and the keto diet itself, because it's so new, I looked for studies specifically on keto, and there are very few. And there was another study like this that was a systematic review where they had tried to find all the clinical trials for keto, and there were so few that were considered high quality that the data, they can't say much yet. But if someone is following this kind of diet, they're avoiding things like this. They're avoiding simple sugars, and this just breaks down for you what that difference is, whether it's 1 or 2. The other side of carbohydrates are complex carbohydrates, and whether if you're avoiding carbohydrates, then you can have some of these, but not others. So there are 2 categories of complex carbohydrates. They're the kinds that are storing energy. Like we said, glycogen is our energy storage molecule in our liver and muscles, and starch is the energy storage molecule for plants. So starch gives you tons of calories because it's just 1 million glucose molecules stuck together. The other kinds of polysaccharides are structural. So cellulose, which is the molecule in plant cell walls, it's the most abundant molecule on planet Earth, is also just made of glucose molecules, but it's linked together in a different way that provides it structural stability, and that's what holds trees up. Chitin is another one that I don't have a picture of here, but it's doing the same kind of work for animals and fungi. So why is it that this is structural and this is energy storage? What's different about them, and why is it that starch gives you so many calories and cellulose does not? This is why. So this is why you don't get calories from fiber, but you get tons of calories from starch. even though these molecules are almost identical. And this is showing it to you in a sugar form, but it's the same exact concept. So even though these long links of carbohydrates might have the same individual pieces, the way that they're stuck together, being flipped back and forth, affects whether or not we can break them down. So energy storage molecules tend to have this kind of linkage, this V shape, and we have an enzyme that can fit into there and break these apart. So when you eat starch, the enzyme breaks them apart, you get a bunch of glucose released. The beta form that's found in structural polysaccharides, we can't break that down. And the only thing that happened is this one glucose flipped around, but our enzyme can't fit to break that apart. And so when you eat something that's got lots and lots of fiber, you don't get energy from it. We can't digest fiber, but Beano can. So folks that take Beano, why would one take Beano? If you don't know about Beano, what is the tagline? Take Beano before and they'll Beano gas. And it's advertised as something that you eat when you're about to eat like a plate of beans or a plate of cabbage or something else that is known to induce gas. The reason why those foods cause gas is because they're full of fiber and they're full of other large carbohydrate molecules that have the same issue where we can't break them down, but sometimes the microbes in your gut can. So if you consume a whole bunch of these things, then the microbes in your gut will start to break them down, except when they break them down, they release gas. And so that gas is now trapped in your intestines, and that's very uncomfortable. So when you take Beano, you're actually taking the enzyme that the microbes have so that your body starts to break those things down beforehand. It means you're going to get more calories from that food because you're going to get more glucose out of it. But it also means that you're less likely to create gas that will make you uncomfortable. So the second thing you read about were lipids. And lipids are unique because they're actually not polymers. So whereas cellulose is literally just glucose stuck together, and you read about the reaction that involves water where you stick them together or pull them apart, right? So lipids don't always fit that mold. What is true about all lipids is that they are non-polar. They are not soluble in water, and that's really how they're defined. It's sort of a catch-all group. So they include fats, waxes, phospholipids, and steroids. And I wanted to click through just a couple of them to show you, talk about them really briefly. You looked at this last week because you were looking at how things cross across the cell membrane. And so this, now you know why the cell membrane is arranged like it is. So this is our molecule. It's got charge up here, which means it can interact with water. And it's got non-polar stuff down here, the fatty acids, which can't. And so it arranges itself in this neat little linear fashion to keep the water out. And that's what gives us this barrier outside of cells. This is our fat storage molecule. So when we consume food that contains fatty acids, those fatty acids will be broken down, attached to a glycerol molecule, and then we get this triglyceride, which is just a glycerol molecule with 3 fatty acids hanging down. And the thing that makes these fatty acids different is their chemical composition. So I brought you this slide, which has examples of saturated and unsaturated fatty acids so that we can just see what the differences are. The key difference is that saturated fatty acids, what they're saturated with is hydrogen. So every place that there's an opportunity to put a hydrogen atom, there is a hydrogen atom. And that means that this molecule is linear. So you can shove a whole bunch of linear molecules together really tightly, which is why they're solid at room temperature. Unsaturated and polyunsaturated or monounsaturated just means how many hydrogens is it missing, have hydrogens missing, and instead they have these double bonds. And so when that happens, you get these bends. So these molecules are not straight because they can't pack close together because they're all bent. That means that they're liquid at room temperature. The last weird group is steroids. So steroids are definitely the odd group because they're not linear. The other ones have these long chains, and steroids are made up of rings. So a lot of folks were interested to read about cholesterol and the fact that cholesterol is so important to sex hormones. So sex hormones, estrogen, testosterone, and progestin are all also steroids, and they are made from cholesterol. So cholesterol in your dietary intake is really important. Somebody was asking posing in the reading yesterday, whether if someone has an imbalance in their sex hormones, there's a role for cholesterol. And the answer is that it's sort of in the opposite direction. So fat levels in the body directly affect sex hormone levels in the body, because sex hormones are fat soluble. So they are produced by fat cells as well. So most of the sex hormones are produced by gonads, ovaries, and testes, but adipose cells also produce fat hormones, which means that the more fat cell mass that someone has in their body, the greater hormone production is possible. And so for some folks, that means that if they have a lot of fat mass on their body, then their hormones might be out of whack because their hormone level is too high. On the other side of that, if someone has a very low percent body fat, then they might have issues with too low of hormones. And you especially will see this in athletes, and it's why so many really, really low body fat athletes who don't menstruate, because they don't have enough estrogen and progesterone circulating in the body to trigger menstruation and ovulation. So fat levels and steroids are really important. The last thing I brought, no, I didn't bring this up the last thing, but related to cholesterol. We talk about cholesterol as in two different ways. So you read about cholesterol, the molecule, which is this thing. But when you talk about cholesterol in the general public, that's not really what people are saying. Because when we talk about cholesterol, we're talking about LDLs and HDLs, right? High density lipoproteins or low density lipoproteins. So I brought you this just to show you what that means and like, what's the difference? How does this affect your body? So we said before that they're not fat soluble, right? These lipids are not fat soluble. So they move through the lymphatic system out of the cells, and then they enter the bloodstream. But once they get into the bloodstream, because they can't be dissolved in water, they ball up, right? So just like your cell membranes are going to clump together to keep water out, the fat cells or fat molecules are going to do the same thing. And so they make these spherical arrangements of lipids where all the tails that are nonpolar are tucked inside and all the polar stuff's on the outside. So these little packets of lipid have different compositions as they move through the body. The first ones that come in after digestion are these really big ones. And they are mostly just triglycerides because that's what you got out of the diet, right? Holding this thing together is also cholesterol, a little bit of phospholipids, and a little bit of protein. And so that's what's in this larger packet. So as this thing then moves through the digestive system, it's going to go to the liver. The liver is the main site of cholesterol metabolism. And the liver is going to pull some of this triglyceride out. So now you can see that these other wedges have expanded because we're taking a little bit of the triglyceride out. As this then leaves the bloodstream, and this LDL is sort of the form of fatty acids for cells throughout the body. And so now its job is this is bringing fatty acids throughout the body. And as these triglycerides are being removed, we see them going down, and it's also picking up cholesterol that is circulating throughout the body. So the sort of VLDLs, we never talk about these, are picking up cholesterol and dropping off triglycerides to create LDLs. So these are the two you've probably actually heard of. So LDLs are the bad cholesterols because they're full of cholesterol. So they're full of the molecule cholesterol, and they have lower levels of these other things. HDLs are the good cholesterols because they have the least amount of cholesterol and triglycerides of any of these packets. And they're called HDLs because they're heavy, because they're full of protein. So these are the two things that are circulating throughout your body. These ones are depositing triglycerides and cholesterol. And the way that is problematic is when that deposition happens inside of blood vessels. So over time, as cholesterol is deposited by LDLs on the blood vessel, that builds up on the inside of the blood vessels, and that causes atherosclerosis, which is hardening of the arteries. On the other hand, HDLs, because they are low in cholesterol and triglycerides, as they circulate throughout the body, they're picking them up. So they're picking up cholesterol and triglycerides to become LDLs. So there's this switch back and forth. These are just swapping. HDLs then are picking up cholesterol, and they can actually move through the body and pick up some of the cholesterol that may have been deposited before. So when someone has high cholesterol, often it's not because of dietary choices. So it used to be thought that if you had high cholesterol, it was because you were eating a lot of cholesterol-rich foods, and that sort of led to this oh, nobody eat eggs, eggs are bad situation that happened in the 90s and early 2000s. We now know that that's not true, that the dietary impact on cholesterol is about saturated fat. So the more saturated fat someone consumes, the greater their cholesterol levels will be. But unsaturated fat and cholesterol itself don't actually make that big of a difference. The largest factor in determining whether or not someone has high or low cholesterol is genetics. So there is a very common condition that most people don't name it as a genetic condition, but it's called hypercholesterolemia. And there are genes known to be associated with this, and it runs in families. So I have this, my whole family has this. You probably know people that have high cholesterol. I was a vegetarian at 25 until they had high cholesterol. There's no way that that was because of my choices, right? So the thing that's happening is that there are differences in this enzyme called lipoprotein lipase. So lipoprotein lipase is catalyzing uptake of fatty acids into the cells, and it, and along with other lipase enzymes, are impacting the composition of these things and how many LDLs are being created, how many HDLs are being created, how often is fat mobilized and moved in these different ways. So that's the thing that like Lipitor acts on. It's acting on these lipase enzymes to affect their activity. So these are three really common forms of dietary fat, right? Saturated fat, butter, delicious. Coconut oil, also saturated, also delicious. Olive oil, canola oil, unsaturated, much less flavorful, still delicious. I went and looked at all the sort of most recent research on these things, and this is what we know. Eating a lot of saturated fat like butter increases our LDL, which is the bad cholesterol, and increases total cholesterol. This is on whole bad. There's nothing good that happens here other than we do need fat in our diet, and you do need cholesterol in your diet. So consuming butter, totally fine. Just don't consume it all the time. Coconut oil also increases LDL, which is bad. It also increases total cholesterol, which is bad. However, it also increases HDL, which is good cholesterol, which is good. So coconut oil is really a mixed bag. It has benefits and it has drawbacks. On the far right, olive oil and canola oil and other unsaturated vegetable oils are good. They are decreasing the bad cholesterol. They're decreasing total cholesterol. Some of them have the ability to also increase HDL and some don't. That's why like olive oil is often touted as being an extra healthy oil, because it can also have impacts on other levels of cholesterol. So if you're choosing forms of fat in your diet, choose more from this side and fewer from that side. What makes different parts of the fat in your body different? Because everyone's heard that like certain types of fat is healthier than others, and that having a pear-shaped body is better than having an apple-shaped body. And so what does that mean? So fat tissue behaves differently depending on where it is in your body. And it can behave differently in all kinds of different ways. So it can be like physically different. It can have higher or lower saturated fat content, but it can also like behave differently by creating inflammatory molecules or not, by responding to insulin or not, and in the way that it releases fatty acids into the bloodstream. So this diagram came from another great paper last year where it was another big review where they were trying to look at everything that we know about ways that fat are distributed and taken in and out of the body. So this is your diagram of someone who has a metabolically healthy fat distribution. And what this means by metabolically healthy is that, like, if you were to do a metabolic panel and you're looking at all of their cholesterol levels and their blood sugar levels and their overall metabolic rate, this is a person who has healthy levels, healthy markers of all of those things. They will have the fat that is stored in their hip and thigh is going to be the predominant fat storage area on somebody like that. And they'll have lower levels of fat stored around the organs and in the belly. This is good because this kind of fat is the fat that behaves well, that does not release inflammatory molecules, doesn't have high saturated fat, responds to insulin appropriately. This is healthy fat. On the other hand, the fat distribution that's often thought to be unhealthy has the flip, where there's a lot more fat stored in the abdomen and around those organs, this deep subcutaneous fat that happens on the belly, and a lot less in the hip and thigh. And so that kind of fat is the metabolically unhealthy fat. And we know that it is doing these different kinds of things to the body that are contributing to not just like overall weight gain or obesity, but also to metabolic health and diabetes and cardiovascular health. These things are impacted largely by, so there are things that we can control and things that we can't. So you can control your energy intake. You can control what kinds of foods you're taking in. You can't control your genetics. And so a large part of this is genetic. On the other hand, your energy expenditure, you can control your physical activity. And to some extent, you might have control of your basal metabolic rate, but also a large part of that is genetic. So fat distributions and the way that your body is going to store fat is just the way that your body is going to store fat. And depending on which way this is happening, determines your risk. Let's talk about proteins. So the molecules we looked at so far, lipids and carbohydrates, are long chains of things, right? They're doing this. What makes proteins really unique is that they fold into an infinite number of combinations of shapes, and because they do so much extra folding on top of just this link in a chain, that gives them variety that no other molecule has. And so that means their functionality is more diverse than any other molecule. So proteins are enzymes, and enzymes are everything. Nothing can happen in the human body without enzymes. Like you would not live for 1 second without enzymes in your body. So just to break these down a little bit more, here's your primary structure, and here are your amino acids. So each one of these has some parts in common, right? It has this gray part where it's got NCC, and then it's got this part on the bottom that's different. So these side chains is what makes one amino acid different from another, and they're what gives what amino acids different character. So we've talked a lot today about polar and non-polar, whether something's water soluble or not. And that's a big part of this. So if this thing, this is often just represented as R. Here's whatever the thing is that's attached. That's right there. If this thing is non-polar, then that means that amino acid can't be dissolved in water. And if you have a protein that's full of those kinds of amino acids, that protein's not going to be dissolvable in water. If you've got one that has a whole bunch of charge on it, electrical charge and really polar things, then it will. And the way that these individual amino acids then fold up together are going to be based on the sequence of what's here and whether or not like this thing can interact with that thing. So at this first level, these are being held together by strong covalent bonds. They don't break apart without an enzyme. You cannot pull one amino acid off of another without an enzyme. Beyond that, the folding that happens is driven by hydrogen bonds. And we talked about this a little bit in the last week when we talked about water, that water hydrogen bonds with other things, and that hydrogen bonds are individually weak, but collectively really strong. So the first level of folding where you get these little spirals, that is driven by hydrogen bonds. The next level of folding where then the spirals are going to fold up on top of the spirals, that is driven by hydrogen bonds and other kinds of bonds as well. And then you read about the fourth level. I don't think I brought you a picture of the fourth level, but fourth level is just when you then have a whole bunch or more than one of these fully folded tertiary levels that then get stuck together. So like hemoglobin has 4 pieces that all get stuck together for it to become a functional molecule carrying oxygen around in your body. And because there's such diversity in the number of amino acids, there are 20 different amino acids. They've all got different sizes and shapes and characters. And you can link them together in innumerable combinations. There's a huge variety of shapes and functions of proteins. And their shape, which is directly related to their sequence, is also directly responsible for their function. So this is one of my favorite ones. I love it because this looks like a butterfly. It sits down on a piece of DNA and slides across DNA. And you can just see how this thing sits down on a piece of DNA and then slides back and forth. This is just like aquaporin. This is another one called porin, right? Where aquaporin lets water molecules flow through a membrane. This is clearly a pore to let stuff flow through a membrane. And then this is one that's fibrous. So collagen is our major connective tissue in animals. This is what holds us all together. Just like cellulose was this long fiber structure that's really strong for plants, this is ours. This is our long fiber, except it's made of protein. So what's important to remember, and this will come up next week for your lab, so we're just sort of introducing this idea now, is that if proteins can fall apart, and when they fall apart, they don't work. And there are lots of different things that can make them fall apart. So we call this denaturation, which is just when the protein unfolds, and it might unfold just a little bit, or it might unfold all the way. The primary structure is maintained, right? Because we said enzymes have to be there to cut stuff apart. But because hydrogen bonds are weak, if you just heat up a protein, it can fall apart. If the pH changes, a protein can fall apart. This is the reason why fever is dangerous, because if your body temperature gets too high, your proteins denature and your enzymes don't work and you die. So when this happens, we maintain primary structure, we disrupt all these other bonds, and now proteins are falling apart. So this happens when we digest things. When we chew food, we break them apart mechanically. And then when it hits the low pH of the stomach, all of those proteins denature because the acidity of the stomach breaks all the hydrogen bonds and they're all linear. Then after they're broken apart, then that's when all these individual enzymes, so here are a bunch of different individual proteases, they're going to come and chop off little pieces. Just because it's interesting, when we cook food, we're denaturing it. And that's why cool stuff happens with your food. So we denature proteins by temperature. So just like fever is bad for your body, it's also bad for your food, except that it makes it delicious. So when you cook an egg, you're heating those bonds, they fall apart and come back together in their own weird way. And that's what makes an egg turn hard. I am a vegetarian. I like to freeze my tofu before I cook it. So you freeze it and then you take it out and you squish it and then you cook it. And it makes it extra chewy and crumbly because you've broken and then reformed hydrogen bonds. pH does the same thing. This is why people like to marinate their meat to make it more tender because it breaks down the proteins. If you've ever had salt cured meat or jerky or anything else, that salt concentration changed denatured proteins, and that's why it changed the nature. And even just like whisking something. So if we whisk air into proteins, that air will physically pull apart hydrogen bonds, and that's why you can make gelatin and Jello. So all of this denaturation means that proteins are falling apart, and for your food, it's delicious. When it happens to your enzymes, that means that whatever that enzyme was doing no longer works. So you're going to come back to this idea next week for lab. But I wanted you to start thinking about, what happens when you perturb some of these situations to the enzymes that are trying to function in that system. So the three kinds of bonds that most people are most familiar with are ionic, covalent, and hydrogen. Okay, so the difference is whether or not electrons are being moved or shared, and in what degree to which they're being shared. So anytime a chemical bond happens, what happens is that there's an electron that's either being moved or shared from one atom to another. So each atom has at its nucleus protons and neutrons, and this is like the heavy part of the atom, and it's what makes an oxygen atom different than a nitrogen atom. And then on the outside of that nucleus, this is where the electrons go, and they're just like spinning around, constantly rotating around the outside in these orbitals around the nucleus. And the number of electrons that are on that outside is really important. It either makes them stable or not. So having 8 electrons in this outer shell makes something really stable. So if there's an atom like this, so this could be chlorine and this could be sodium. If there's an atom like this that's missing 1, it has 7 out of 8, it's really, really drawn to electrons. It really, really is going to attract electrons because filling that last space is the whole thing that all of its energy is directed towards. This atom would much rather get rid of this one because it's hanging out by itself. And underneath this shell, there's another shell, and the one underneath that's complete. So it would like to get rid of this electron. So what happens in an ionic bond is that one atom transfers the electron to another physically. And that means that this one has lost an electron and is now positive. This one has gained an electron and is now negative. And the thing that's actually holding them together in an ionic bond is magnetism. So they are stuck together like magnets. In a covalent bond, instead of transferring from one to another, they're going to share. So both of these atoms only have 7. They both want 8. So instead of one giving it to another, which would not solve both problems, if they share, then now each one of them has 8. So that is a covalent bond. So these electrons are spending their time circulating around this one and this one pretty evenly. If that's true, then that means that neither one of these have any charge. The atoms are neutral and the bond between them is neutral. So that's what happens like in lipids, all of those carbon and hydrogen bonds, those long chains, they're behaving like this. A hydrogen bond happens when you have a covalent bond that is not sharing equally, where one atom is really hogging that shared electron. So that, like, let's say if this is oxygen, oxygen is a complete electron hog. So this little shared electron here, it's technically shared, but it's like spending 90% of its time over here. So that means that this molecule has a partial positive charge and this one has a partial negative charge. Or sorry, other way around. This one has a partial negative because it's getting the electron more. And that's what this little designation down here is showing you. So here's an example where here's a hydrogen, it's getting its electron stolen. It's got a partial positive charge. Here's the oxygen that's, you know, totally stealing this electron that it's sharing. So it has a partial negative charge. And when those things exist, the magnetism between this molecule that has that and this molecule that has that, that is a hydrogen bond. And so this is what's happening on proteins. If we go back to this, if we go back to this, then here's an example where this has a partial charge and this has a partial charge because their covalent bonds are shared unequally, right? This is an oxygen, that's a hog. And so because these have partial charges, they can stick together to each other. So it's also magnetism. It's much weaker than the ionic magnetism because this is a full charge, a full electron, and this is partial.